Sorry for inconvenience. So today I am very happy to introduce my Guru Bhai, Professor Santosh Kumar. We both worked under the supervision of late Professor Umakant Sinha during our PhD program. He is very senior to me, and I am thankful to him because presently he is not in India. He is presently in Hong Kong, and the time difference is 2 hours 30 minutes. So uh, I am very thankful to Professor Kumar. So he agreed within a very short notice to deliver a talk uh, to you people. Before the session, I would like to give a brief introduction and achievement of Professor Kumar, former professor and head Department of Botany and Dean Faculty of Science, BRA Bihar University, Muzaffarpur. He did MSc from Ranchi University and PhD botany PhD botany with a specialization in microbial and molecular genetics from Patna University in collaboration with NCL, National Chemical Laboratory, Pune. He also served as Professor of Biotechnology, Amity University, Noida from 2002 to 2006. He also served UGC Coordinator, Special Assistance Program, DRS Phase 1. He has published more than 40 papers in both national and international journals. He has successfully guided 21 PhD students. He has successfully completed many research projects, and he travel a lot uh, because I am giving some of the his guest lecture and talk in international fora. For example, Mendel Genetic Conference, Brno, Czech Republic, KTH Royal Institute of Technology, Stockholm, Sweden, uh, Masank University, Mendel Museum, Brno, Czech Republic, Upasla University, Linnaeus Museum, Sweden, Nobel Museum, Stockholm, Sweden, University of Helsinki, Finland, John Innes Institute, Norwich, UK, and Maastricht, Amsterdam, Netherlands. He also got Gajale Memorial Lecture Award by Indian Botanical Society in 2017. He also served and is still serving as a member of many scientific academic organizations like Member Scientific Committee, TPSC Agartala, Member Selection Committee, GT University Ahmedabad, Member NAC Peer Team, Bangalore. So I am very thankful to you, sir. So now you can start your talk.
Hello,
हाँ आ रही है
uh,
आवाज.
very good morning to everybody, though here it is afternoon. And I am thankful to the organizer and present head of the Department of Botany, Dr. Birendra Prasad. You know, he has elaborated my achievements, that to some extent it is not that way. Again, I start with the second lecture, second slide. It is, you are just saying, learning from to tomb, from birth to death. Actually, when Sridhar asked me to deliver this lecture, I immediately accepted because I realized the words of Professor Goodengod, who got Nobel Prize for lithium ion battery in 2019. When somebody asked him, "You still work?" Because he was working, he got the Nobel Prize at the age of 97 years. So he commented, if one superannuates, it does not mean he or she should wait only to die. So I take this privilege and I always like to deliver. The topic next, the topic which I proposed, genetics of mitochondria and nuclear DNA with respect to Neanderthal ancestors. Neanderthal, Dr. Pabo got Nobel Prize because of the DNA sequencing of our ancestor, that is called Neanderthal ancestors. In Germany, Thal is used for cave, and the place name is Neander. So the ancestors were called of that period Neanderthal ancestors. One thing very important, if you write Neanderthal with capital N, it will be the ancestors. But if you write Neanderthal with a small letter n, then it will be an abuse by the Britishers. You may, if he says you are idiot, it means he is using Neanderthal with a small n. Next slide. I have compartmentalized my lecture into three headings: genetics and me, DNA, that is two source, mitochondrial and nuclear, and the third is Neanderthal ancestors. Next slide. Genetics and me. When I was a student of interclass, my one of the classmates remarked that particular teacher is going there. Are you related? Are you his brother? I retorted, because I was told that not to disclose that my brother is there. So actually what happened, on my retaliation, he clarified that because you two walk in the similar fashion if seen from the back. And this is the first lesson of my genetics, because in reality he was actually my elder brother. Next slide. Research, as you people may have done your research work or likely to enter into the research period, you will realize that it is not always positive, because years of research period, when I faced 1984, I was supposed to work on plasmid of a bacterium, Thermoactinomyces vulgaris. But the failure was there. Unless I went to NCL to find that in my organism, plasmid was not there, but amplifiable unit of DNA was the main source, who used to give 24 kbp band, which we initially thought to be a plasmid, but it was not the plasmid. So after so many failures, we got some positive conditions in my bacterium. You know, 1850s, that is middle of this 19th century, it was Gregor Johann Mendel, who has no knowledge of gene and DNA, but he predicted whenever a hidden character would appear in the following generations. We all know all the three laws that he formulated. Recently, in July 20 to 23, 2022, after my retirement, I attended the bicentennial birthday celebration of Gregor Johann Mendel at Brunn, Czech Republic, and I treated this journey with pride, as it is my highest pilgrimage. I may like you to share a particular clipping for 2 minutes. Perhaps you may not hear the clippings. So next slide, please. Mendel actually sent all his work to Darwin to get some clarification. But Darwin put the paper in a drawer without giving a thought. Perhaps his action was out of frustration, what we call jealous approach towards Mendel. Darwin theory, based on evolution by natural selection, we all know we have so many pros and cons of this hypothesis, because it is nature rather than human judgment that determines which organism live long to have offsprings. Next slide, please. We are tending towards molecular genetics and genomics in 20th century, a study of how living things give their offsprings the instructions or genes for a particular character. These particular features came into existence when the DNA was declared as the genetic material by Avery, MacLeod, and McCarthy, followed by Hershey and Chase. Then the recombinant DNA genetics came, and genomics and proteomics, other branches of genetics, started, which indicate nucleotide sequences in DNA and amino acid sequences in protein. Next slide, please. What is the impact of mitochondria? The second compartment I am discussing. The impact is in mitochondria, the churning of food converts into energy. You know, ATP synthesis that we are observing in mitochondria, it is being helped by mitochondrial DNA. Because if we trace the history of mitochondria, we are likely to suppose that mitochondria have originated from free roaming bacteria. These bacteria were eaten by prokaryotic cells, eukaryotic cells rather, but it has not been digested by the larger cells. And after millions of years, these bacterial cells became mitochondrial in nature, became friends, and are inseparable. Next slide. Recently, I saw some adjective for Y chromosome and mitochondrial DNA. As you have seen, as you are seeing in the slide, Y chromosome has been given the adjective of Adam, and mitochondrial DNA has been given the adjective of Eve. Why it is so? If you remember, Y chromosome pass unchanged from father to son. That is, we males get this Y chromosome from our fathers, who gets from his father, that is grandfather. Again, grandfather gets from great grandfathers, and this system continues. Similarly, mitochondrial DNA, which a female inherits from mother inherits from her mother, that is grandmother, again grandmother to great grandmother, and this process continues. Since exclusive donor is mother in case of mitochondrial DNA, the adjective has been given as Adam, as Eve, and similar approach given to Y chromosome as Adam. Next slide, please. Mitochondrial DNA, theoretically, every one of us has inherited his or her mitochondrial DNA from our mother, from the female who lived long ago. But with the passage of time frame, some sort of alteration were invoked, of course, through mutation. Credit for such alterations was given to the random mutation, which was altered the genetic code in million years. Next slide, please. So, what is my contribution to mom? We are thankful because she has given her egg cell in our constitution. Actually, what happened? Everybody knows that during fertilization, egg cell attacks and destroys sperm's mitochondria, so that the latter fails to contribute any mitochondria in the developing embryo. Actually, a sperm enters into a tract, and in that process, it requires ATP. So naturally, all its ATP being consumed in the process of movement into in the vicinity of egg. Thus, every cell in our body has mitochondria, and all of them are similar and are descendants of mitochondria that our mother gave to us. Next slide, please. The mitochondrial genetics, if we concentrate, it is a single circular present in mitochondria, and it can divide any time the cell likes, irrespective of cell division. Cells are believed to retain hundreds, even thousands of mitochondria. Sperms also have mitochondria, but as I told you, that in the traveling process, when it travels from in the vaginal tract, it's before reaching to egg, it all mitochondrial are exhausted because of the energy that it requires in the traveling. Next slide, please. As per Human Genome Project, we have the genomic DNA, as you remember, 3.2 billion base pairs per haploid. So if you consider the diploid cell, it will be 6.2 billions. But mitochondrial DNA has been seen with only 16,500 base pairs. So if you calculate that way, it will come to 0.0005% of the genomic DNA. See the diversity between our cellular DNA and mitochondrial DNA. Since thousands of copies of mitochondrial DNA of just one cell, two copies of our mitochondrial DNA are present in thousands as respect to two copies of genomic DNA in our cell. So by 1996, thousands of mitochondrial DNA have been sequenced throughout the world. Next slide. What are the genes present in mitochondrial DNA? You see, the circular mitochondrial DNA is without histones, and it has, as I told you, 16,500. Actually, the number has been disclosed 16,569 base pairs. It contains 37 genes. It codes 2 ribosomal RNA, 22 transfer RNA, and 13 polypeptides for protein as per sequencing data. One thing is very important. You see, the sedimentation constant of the ribosomal RNA are 12S and 16S. The presence of 16S ribosomal RNA in ribosomes, mitochondrial ribosomes, indicates its nature to be bacterial. These mitochondrial DNA encoded polypeptides are all subunits of enzyme complexes which are required for oxidative phosphorylation in mitochondria. If you dissect the mitochondrial DNA, if you separate the mitochondrial DNA, you can have two strands. If G and A are present in one strand, it will be heavy. And if T and C are maximum in another strand, it will be light. Next slide. I am presenting before you the organization of mitochondrial genome. The organization of mitochondrial genome, as you are looking, it has the origin of replication, single point for origin of replication. It is again bacterial properties, and 2 ribosomal RNA genes, 22 transfer RNA genes, and 13 protein sequences are there. All has been chalked out. And actually, what we require, 16.5 kb of mitochondrial DNA that has encoded 2 ribosomal RNA, 22 transfer RNA, but 13 of 83 genes. I have given you the different genes, 13 genes in this slide, because the mitochondrial DNA contribution is, if any mutation occurs, because mitochondrial DNA units have accumulated in enormous number, so there is no effect of mutation which can cause disease in offspring. So is the basic properties of this mitochondrial DNA. Heteroplasmy. Actually, when mutation occurs in some of the mitochondrial DNA, the rate of mutation is quite high. Why? Because the free radicals which are released during oxidative phosphorylation, they can damage mitochondrial DNA. And it cannot be repaired because the repair enzyme system is not present in mitochondria. So how it can save itself? Again, it is the enormous number of the mitochondrial DNA that will not allow the mitochondria to get the mitochondrial DNA to express any of the mutants. Mutation in one or a few may not be expressed in presence of hundreds of copies of normal mitochondrial DNA, which are present in either a skin or muscle cells. Coming to the genetic properties of mitochondrial DNA, you will notice so many variants. And when the mutated form of mitochondrial DNA live along with the normal DNA, this coexistence is known as heteroplasmy. In this, both types of mutated form and normal form live together inside the mitochondria. Next slide. Again, the bacterial properties you can observe in mitochondrial DNA in respect that there is no intron and only one replicon, which enables massive piece of RNA, synthesis of massive piece of RNA. Further, it cuts into several pieces, and that will act as either transfer RNA, messenger RNA, ribosomal RNA, which are required for protein synthesis or polypeptide synthesis in mitochondria. Next slide. Mitochondrial DNA-based sequences and amino acid sequences of the corresponding protein indicate that genetic code is different. 4 out of 64 codons have different meanings from those of the same codons in universal, if we consider our genes. Different genetic code AUA codes for methionine in mitochondria, which normally codes
Kindly change the
isoleucine.
slide, please. It's not in the sync. Sir is explaining the next slide, maybe.
Variant genetic code. The slide I am talking variant genetic code. Has it come?
It's actually one slide lagging.
No, it's not in the sync.
Oh, variant genetic code is not there. Anyway,
No, sir.
see this mitochondrial genetic code.
No, sir.
Is it
Now we are
Yes,
on
sir.
variant genetic
Yes,
code,
yes,
sir.
yes.
is it there? Mitochondrial genetic code.
Yes, sir. Yes, sir.
Now, so variant genetic code is there. You can continue now.
Oh, okay. So what I am speaking that AUA in mitochondrial DNA codes methionine, whereas it normally codes isoleucine. Similarly, UGA codes tryptophan, where it is a stop codon. Similarly, AGA and AGG. Normally, it codes arginine, but in mitochondrial DNA, it works for a stop codons. I have given you the whole scenario in this. Next slide, please. Mitochondrial genetic code. You can see can you see?
Yes, sir.
Universal,
Yes, sir.
universal code. You see the 4
Yes, sir.
codons in mammals and in the mitochondrial DNA. We see that AGA, AGG acts as a stop, whereas it is meant for arginine as a universal code. Next slide, please. Ovule positions. Like normal, we have several ovule positions in the mitochondrial DNA. Since the normal codon-anticodon pairing rules are relaxed, we have the normal codon-anticodon relationship as a ovule condition, but in mitochondria it is relaxed in the sense that many transfer RNA molecules recognize any one of the 4 nucleotides in the 3rd position, permitting protein synthesis in mitochondria because the transfer RNA are few. However, more than 99% of mitochondrial protein required for maintenance, replication, and transcription are encoded by nuclear DNA and later on transported in mitochondria. Next slide. So we are observing dense gene packing. Since no introns in mitochondrial DNA regulatory genes are there, that resulted in a long RNA transcript, and then it gets clipped to form 2 ribosomal RNA, 22 transfer RNA, and 13 messenger RNA. Absence of introns obliquely supports mitochondrial DNA connected to bacteria, as I told you. In yeast, what we have observed, that is a different thing. Introns seem to be in and out in the genome of yeast, giving the origin of transposon. That will be the talk in any of the next lecture. What are the advantages of this mitochondrial DNA? Scarcity and deteriorated samples, or when biological material is limited, this mitochondrial DNA will be of immense use in forensic science. I wish to inform you that Government of India is going to have number of laboratories for forensic test. I was recently in Agartala to have an interview in the interview board, but I didn't find a very good person of forensic science background. So I will advise my colleague to expose this forensic science to students to get enrolled, have the degree in forensic science. It will provide ample opportunity in the coming days in India. So useful material line of inheritance or maternal ancient ancestry, since the cytoplasm is controlled only by egg during fertilization. Next slide. Now coming on the third part of my lecture, that is Neanderthal Genome Project. This project was able to recover enough DNA from a bone believed to be 50,000 years old. And it was found, and later it was found during DNA analysis that Neanderthals were 99.7% similar to present day, that is we, Homo sapiens. And that every Europeans, it has been calculated, may have inherited at least 4% of their DNA through Neanderthal ancestors. On comparing with Neanderthal mitochondrial DNA, changes in sequence of thousands of human mitochondrial DNA were noticed. So the person who got the Nobel Prize, Svante Pääbo, what he did, he was confused. He was thinking that some cow mitochondrial DNA has amalgamated. So he again undertook the sequencing of cow maternal DNA, and later he was convinced that cow mitochondrial DNA was not there. Neanderthal disappeared some 30,000 years ago. They were considered to be the closest extinct relative of contemporary human than ape. Till now, we were thinking in terms of our close relative to be ape. But with this discovery, because human differed chimpanzee a bit over 1%, that is 99% sharing, whereas with Neanderthal, we are sharing 99.7% homology. Next slide. The oxidative phosphorylation, for which the system is creating such type of mitochondrial DNA. So oxidative phosphorylation being carried out for generating ATP is the only biochemical function that is controlled by mitochondrial DNA, whereas other functions are regulated by nuclear DNA. mitochondrial DNA codes for some protein, subunits of electron transport chain, ribosomal RNA of mitochondrial ribosomes, and other proteins. Next slide, please. There is one very important aspect, that is damage that occurred in our DNA versus repair mechanism. Deamination is a normal phenomenon that we always encounter. It results cytosine into uracil if the amino group is removed and replaced with correct nucleotides under repair mechanism. The uracils which have been discarded remain as cellular garbage, and it is excreted in urine. Thus, repair system at the cost of ATP synthesized in mitochondria. This synthesis of ATP in mitochondria helps and prevents DNA from any damage, because the joining of new nucleotides requires ATP, and that ATP being given by mitochondria. When we die, in our dead body, the dead body cannot breathe, no aerobic respiration, naturally no ATP synthesis. And since ATP is meager, synthesis has been checked, so repair system also got blocked, and that resulted into the damage of our DNA. Next slide. The person Svante Pääbo, he is a Swedish scientist, and he got Nobel Prize in 2022. Succeeded in sequencing the genome of Neanderthal. At that time, I was in Sweden, and I shared by visiting the Nobel House in Stockholm that he discovered hello, Svante Pääbo. Hello, slide is coming.
Coming.
Yes, sir.
So he found that gene transfer had occurred. He found that gene transfer had occurred from these new distinct hominins. Hominins is a group which represents present human group, the ancestors, as well as Neanderthals ancestors. So this Homo sapiens, following the migration out of Africa, it has been estimated some 70,000 years ago. Next slide. On comparison, we find actually what Pääbo did. He took 379 nucleotides of Neanderthal mitochondrial DNA and compared with 2051 bases of present day human. What he observed, that there is no relationship between the mitochondrial DNA of Neanderthal and the mitochondrial DNA of present day human. Since there were mitochondrial DNA of Neanderthals differed at 28 points, whereas in our cases, we have the difference of say at 7 points. So this demarcation made us to make a mitochondrial phylogenetic tree, and the present day human trace their ancestry back to one common mitochondrial DNA ancestor to whom we have called mitochondrial Eve. If you look upon this particular ancestor tree, mitochondrial DNA tree, you see the 4 Africans, the African Americans, the Africans, non-Africans, they all have arisen from a red slot, red point you are observing. That is the mitochondrial Eve. And the non-Neanderthal.
Move slide. Sir, please move slide.
This slide is tree, phylogenetic tree. Did you get that?
Yes, sir. Yes.
So all these present day humans, they got it from this red point, and they have been bifurcated. So they have the similarity and dissimilarity with Neanderthals. Based on mutation rates in closely related species such as humans, Neanderthals and apes, Pavo estimated mitochondrial DNA ancestors of present-day human. The mitochondrial Eve lived between 1 lakh to 2 lakh years ago, whereas Neanderthal DNA lived about 5 lakh years ago, which means that Neanderthals were 3 to 4 times as ancient to today's human. All these facts led Svante Pääbo to prove that mitochondrial DNA different from Neanderthals. Next slide, please. The gene flow of individuals in and out of a population inducted new alleles, thereby increases genetic variation. The ancient flow of genes to present-day human has some physiological relevance, as for example, the immune system, which reacts against infection. We developed the immune system with the passage of time, and perhaps our ancestors, they didn't have, and that may be a factor for their extinction. Next slide, please. An extinct species of human widely distributed in Ice Age between 1,20,000 to 35,000. Looked upon a human with a receding forehead, but had more prominent brows, protruding faces, larger eye sockets. That I will show you in the next slide, how they look. Next slide. Next, taxonomic nomenclature. Did you get that? Taxonomic nomenclature. 2 slides ahead.
It's not. No, it's not. No, sir.
Next slide, next to next. I have crossed 2 slides.
Yes, yes, sir. Yes, this one.
You know, this was named as Homo neanderthalensis. Neanderthalensis. But somebody, again subgrouping Homo sapiens neanderthalensis, as for example, we call ourselves these days Homo sapiens sapiens. Next slide. See the reconstructed skeleton, which I have taken from, with permission. Next slide. American Museum of Natural History. You see the difference between the pelvic region and broad chest, the heads, the eyes. So they are, have you seen the, are you looking there on this slide?
No, sir. Once again, the previous. Yes, yes.
No,
No,
sir.
next.
No.
Skeleton. Did you get
Yes.
that?
Sir.
Yes, sir.
Yes, right now. Yes.
So this reconstruction of a skeleton has been done by, and I have taken with courtesy American Museum of Natural History. You see the comparison. On left side it is Neanderthal, and on the right side the present day human that we are. So Pääbo, what came out from the next slide, please. Next slide. Neanderthals are genetically distinct from modern humans, but are more closely related to us than chimpanzee. The Neanderthals are modern human lineage diverged about 5,50,000 years ago. And there is no evidence of Neanderthal mitochondrial DNA lineage in modern human. Actually, what was the concept that was given by Pääbo? That if you make a cross between Neanderthal male and present day and human female, the progeny is fertile. And if the progeny is fertile, it will carry mitochondrial DNA of Neanderthal females. But if in opposite crossing, that is, you take female of the Neanderthal and male of the present day female, and if the progeny is infertile, there will be no further transfer of mitochondrial DNA. That's why Pääbo made the hypothesis that though there is similarity in the nuclear DNA, but there is no trace in the mitochondrial DNA of present day humans compared to Neanderthal males. Absence of genetic diversity and evidence of incest ensure a smaller population. Incest means, if the crossing is being done with close relatives, since they predicted that their offsprings came from the between the meetings of close relatives, that may lead to their extinction. This is a particular full graph. You see the Homo sapiens just far away from the Neanderthal. So this graph indicates that evolution has taken place. The present day human, which we were still believing 1 lakh year, actually it came in 30,000 to 40,000 years ago. And ancestral ancestry that in Neanderthal is the immediate ancestor that Pabo, through his sequencing method, which he did, he was very hopeful of. He has been very doing on PCR, and he was very thankful to the discoverer who got Nobel Prize for the discovery, for the invention of PCR. So this is what I want to express before the audience, that Homo sapiens, how they came out in the present situation. Now I am ready to, if you have any question, because time is running short. Time is still there? Hello? Hello?
Yes, sir. Yes, sir.
Yes, sir. Yes, sir. Half an hour at least.
हाँ स्लाइड तो आ रहा है
After 2.
आपका.
Now coming on the fourth part of my lecture, that is nuclear DNA. Nuclear DNA, beyond doubt, everybody knows, we have come to convince this, that DNA proved to be a genetic material which transmits the characters from one generation to next generation. We resemble parents, we resemble grandparents, we resemble our maternal uncle, but not maternal auntie. We resemble our maternal, paternal, what we call bua, but not Fufaji. Why? Because the distribution of DNA is like that. So, what I want to impress, that lineage of 7 generations. Suppose I got my DNA 50% from my mother and 50% from my father. If you go to the back generation, that is, my father has contributed 50% and he got 25%. So we got 25% from our grandfather, 12.5% from our great grandfather, 6.5% from that next back generation. So if you go downwards, if you go like this 7 generations, the lineage will tell you that we are getting 0.7% DNA of our 7th generation parents. May I cite you one example which is prevailing in Darbhanga, that in Brahmin's family, up to 7 generations, if it is traced, if that pedigree analysis suggests that the bride and bridegroom belongs to below 7 generation, the marriage cannot take place because of this fact. So our progeny of 7 generations, similarly we have been getting, we can go for marriage in 8th generation, and the 7% DNA, which is just negligible. So up to 7 generations, there is no question of inbreeding. The dogma, which we call principle, which is universal in every sort of life, whether plants, animals, human beings, bacteria, even viruses. Now coming to the probiotic molecules. Next slide, please. Carbohydrates, proteins, and nucleic acid. I am referring the 3 slides which I have taken from molecular biology of gene. Can you get this synthesis of amino acid, the slide for synthesis of amino acids? Is it before you?
Yes, sir, it is.
Yes, sir.
This is for one reason. What I want to impress, you see the reaction. Ammonia plus nitrogen plus water, and in electric discharge, which occurs in nature, produced hydrogen cyanide and aldehyde. This aldehyde with ammonia, when reacts, gives a number of amino acids. Next slide, please. That formaldehyde, aldehyde that is being produced in last slide, can polymerize in order to form ribose. Next slide, please. That hydrogen cyanide, these oligomers polymerize and can produce adenine. Adenine changes into uric acid, which can give rise to uracil. What I want to impress, that these adenine, thymine, cytosine, uracil, and guanine must have been produced on Earth, because I am presenting before you a prebiotic origin of life. Please concentrate on this particular slide, and I will advise my fellow colleagues must teach this to your students. You see, is the slide is before you, prebiotic origin of life?
Yes.
Hello.
Yes, sir.
This is very important slide.
So
You
there is
see,
synthesis of unit of
from the
nucleic acid.
Yes, sir.
yeah, after synthesis, I have come on prebiotic origin of life. Please concentrate on this particular slide.
Now it is
You see from the lower end, prebiotic molecules. These prebiotic molecules, you know, we have always come to know that a piece of sun which has been rejected, and after so many years of condensation on the Earth, came out in our system, in our galaxy. So the year was 4.6 billion years when the Earth came. You go upward, electric discharge, and in presence of UV light, as I have shown you, different chemical reactions took place, and sugars, amino acids, and nucleotides were synthesized. As I have shown you in my previous 3 slides, how the nature has synthesized sugars, that is carbohydrates, amino acids, and nucleotides. These sugars metamorphose into carbohydrates, amino acids polymerize into protein, and nucleotides, adenine, thymine, cytosine, guanine, which has been constructed in the previous slide, they form nucleic acid. I want to compare a fisherman. When the fisherman throws his net, what happens? The outer boundary of net represents cell wall. Outer boundary of net represents cell membrane, which is of protein. Inside, some stones, if being captured, they will represent carbohydrate, that is for metabolic process. And if fish is being captured in the net, I think it represents genetic information system. So first prokaryotes has been originated due to the formation of membrane, metabolic, carbohydrates, and genetic information system. And this first prokaryote originated when 3.5 million years ago. Now, billion years ago. Now you see, Earth came 4.6 billion. First prokaryotes came 3.5 billion. So the gap is 1.1. Today, people are discussing that we expect life in Mars. So here, the transition period of 1.1 billion years, can we think that the same that Mars is also passing through this period, this intact period, and after few billion years, after few years, 1,000 years, the life we can observe on Mars. Then came first prokaryotes, multicellular organisms. The question is, what is the proof? The proof that Miller with his counterpart Urey and Miller both did this experiment. Next slide. Here I have taken it from your basic books. You see the boiling water and all the natural gases were placed where electric discharge was made. And after condensation, the amino acids, the bases, nucleotide bases, and sugars were formed. Next slide, please. You see the photograph of Miller of his laboratory where he constructed that possible experiment. Urey and Miller, Urey awarded Nobel Prize, next slide, for deuterium discovery. So the Earth primordial atmosphere, hydrogen and hydrogen containing gases in ocean. However, there is no molecular oxygen. Water was there, but molecular oxygen was not there. Next slide. So my brief idea is to impress origin of life, that is electric discharge, stimulating the UV radiation, which was introduced in the primordial soup, where glycine, alanine, glutamic acid, valine, proline, other amino acids, along with sugar and nucleotides, were synthesized. I hope I have covered different aspects related to. Hello, next slide, please.
Yeah, we have come to the end of
That
end slide.
presentation.
Yes,
So
yes.
thank you to all. And I, as usual, I also thank DNA.
So the session is open for, we have
question.
only 15 minutes. Next session will start at 12. So few questions may be asked from Professor Kumar.
Okay. I am really sorry that some disruptions are there and I could not fully.
Thank you.
Actually, though there is no problem in the connection, but somehow the technical situations came and it makes some delay in the presentation. Any question?
Sir, as you mentioned about the Brahman family in Darbhanga,
Yeah.
can you tell some more things about that?
Actually, the marriage which were being held in the Sabha Gachi, when the two group comes for marriage, they will go to pedigree analysis. And if the situation comes that they are cousins of fourth generation, the marriage will not take place. So genetically, what I want to impress, that genetically it is also correct. So in breeding, as the Swanto Pabo told, that the death of these Neanderthals ancestors happened because of the inbreeding. And they died, but we survived. Because our genome have the variations, but their genome's lineage was not of that variation to compete with the nature, compete with the atmosphere of that time. So it is very helpful, and genetically it is correct that inbreeding is not supported. Thank you.
Thank you, sir.
Good afternoon, sir. I have a question. Good afternoon,
please.
sir. Yes,
Yeah,
sir.
please.
First of all, thank you, sir, for such a wonderful session. Sir, in the presentation, you discussed about the mitochondrial DNA nature. So you told that the double-stranded DNA, one strand is generally heavy and another is lighter. Like one is purine rich, another is pyrimidine rich. Can we impart
Yeah.
any significance to this?
Yeah, because in the PCR analysis, what Swanto found always a heavy band. So later he found that it is due to the presence of more guanine and more ATP, adenine. Because in the corresponding DNA thread, naturally it will be pyrimidine. So since purines are two-ring structure and pyrimidines are single-ring structure, so their results came as heavy and as the light chain in their autoradiography. So that was the solution that he got in the later analysis of DNA sequences.
Sir, does it provide any molecular significance to the plasmid DNA having one strand lighter and one
No,
heavier?
no. Since they were getting two bands in the autoradiography, so that suspicion started why such differences are there. So in the mitochondrial DNA of Neanderthals, they've got two bands of different heavy and light, and the regions were obscure. So he, on the detailed description of the DNA analysis, then they found that one coming with purines and other naturally coming with the pyrimidines.
Okay, so regions are obscure yet. Okay,
That
thank
may
you.
be the reason that the obliteration happened with the Neanderthals and the composite relationship was not maintained as we are maintaining these present day humans.
Thank you, sir. Thank you so much.
Sir, I have a question.
Thank you so much, sir.
Sir, in one of the slides, you said that in the experiment, he took 379 base pair of Neanderthal mitochondrial
Yeah.
DNA and
Yeah.
2051 base pair of present day human.
Present human. They compared.
So why didn't they compare the complete sequence? Like, this is very not very uniform. 379 base pair of the Neanderthal and
I tell
2000.
you, the DNA that they isolated from Neanderthals were in fragmentation because the damage was there. Uracil, if being replaced by thymine, the cytosine deamination results cytosine into uracil. The uracil will be taken out by the repair system. So what happened that the damage was done in the isolation of the mitochondrial DNA. So he took the pieces of mitochondrial DNA pieces and he got one piece of that 379 nucleotides and compared with this, and what he found that maximum variations was present in our system, not in that from the different fragments that he considered in the PCR. So yeah.
Thank you, sir.
सर एक चीज सर को प्रणाम, मैं दिलीप बोल रहा हूँ सर, पटना यूनिवर्सिटी जूलॉजी से सर। आपसे मुलाकात है पहले से भी। सर एक चीज हम लोग बहुत कॉमन ये करते हैं कि मुस्लिम फैमिली में बहुत क्लोज रिलेशंस में शादियां होती हैं सर, बहुत क्लोज रिलेशन में। इवन द कजन्स आर गॉट मैरिड। ऐसे केसेस में जेनेटिकली कितना करेक्ट है ये सर, और क्या ये
नहीं करेक्ट है? मैं आई गिव यू आंसर। In the Muslim family in which I am very close, I didn't allow them to marry in the cousins, because if you take the system, if you take the chart, death more in the Muslim families of the fetus than in Hindus because of this reason. So in the family where I am close that way, I put the pressure not to have the marriage between the cousins, among the cousins.
Sir, in Europeans and British people also, they used to maintain those pure bloodlines. In Europe and Britain, they used to maintain those pure bloodlines where they don't let any non-royal blood enter the royal family.
It happened. So, you know, so many diseases that they carry, even in the case of Maharani, of Elizabeth, you know that famous disease that in the family was carried. So this inbreeding is highly objectionable. And always you go for, even in the Hindu system, no marriage takes place if the gotra is the same. I remember so many cases, the marriage has been settled, but once they came to know that the same gotra prevails between the two groups, the marriage became cancelled. So it is always better to have the marriage between the distant relatives, distant persons,
Sir,
unknown
I have
persons.
a small question.
Yeah.
Sir, as you have told that Neanderthal has extinct due to inbreeding.
Pardon?
As you have told that Neanderthal
Yeah.
man extinct due to inbreeding.
Yeah, inbreeding.
So how it can be possible to analyze mitochondrial DNA and conclude upon that Neanderthal has extincted due to inbreeding?
You know, the mitochondrial DNA only mother gives. So that's why I give the name Eve to the mitochondrial DNA females, because that DNA will never get amalgamated except mutation. That will remain as such. But with the passage of time, mutation you cannot check, mutation occurs, and that will change some of the nucleotides in the sequence. So far, the maternal mitochondrial DNA is concerned, as I told you in one of the slides, that if the marriage takes place and there is no daughter in the family, so the mitochondrial DNA will vanish of that family. Similarly, if you think of Y chromosome, I am facing the same problem in my family. We were 5 or 6 brothers having only one sister. But in my son generation, my next generation, there is no child, male child. So in the family of my family, the Y chromosome going to elope, going to be vanished.
Sir, I have one question, sir.
Yeah.
Sir, you have thank you, sir, for such an informative lecture. And sir, you have mentioned about the origin of life.
Yeah.
Sir, is that what is the first sign of life? Is that the formation of amino acid in primordial soup?
No, I told you, I have shown you the 3 diagram chemical reactions. That is the amino acid, methane, hydrogen, water vapor, under electric discharge, they form. And that was experimentally proved by Urey and Miller. That I showed you. That after the reaction they got, after condensation, they got a few amino acids, they got few nucleotides and sugars. So the 3.
Okay, sir. That is. I got my point. That is the first sign of life is the formation of amino acid. Then life is produced from that.
No, the amalgamation. You know RNA enzymes.
Yes.
When I went to the Mendel celebration, that gentleman who got the Nobel Prize for RNA enzymes also came, and I got that opportunity to have a discussion with him also on this particular point. So amino acid synthesis, carbohydrate synthesis, initial sugars and nucleotides, they were all synthesized simultaneously in the soup, prebiotic soup. That we call chemical evolution. So biological evolution, chemical evolution always precedes biological evolution. Chemical evolution, we got all types of nucleotides, all types of amino acids, and polymerization occurs at high temperature. Polymerization occurs if there is no enzyme, high temperature can be suitable for polymerize these molecules. So somehow the proteins were synthesized, enzymes were synthesized, and this nucleic acid were synthesized.
Sorry to interrupt you, sir.
Thank you, sir. Thank you.
I now request Dr. Meena Bala to give a vote of thanks to Professor Santosh.
Yeah, okay. Thank you.
Am I audible?
Yeah, audible.
Good morning to all. On the behalf of all the participants and the organizing team of this course, it is my privilege to propose vote of thanks to such a distinguished and experienced academician, Professor Santosh Kumar. Sir, we extend lots of gratitude and respect to you
for sparing your invaluable time, for sharing your own research experiences with us and enlightening us with the knowledge of prebiotic origin of life, genetic principles behind the application of specific regions of mitochondrial DNA and nuclear DNA to study human evolutionary biology, and importance of and important findings on the relationship of evolutionary relationship between Neanderthals and Homo sapiens. Thanks to all the participants for their support and active participation during the lecture and Q&A session. We are highly obliged to Dr. Atul Pandey, Dr. Virendra Prasad, and all the members of the organizing team for putting their best efforts for inviting such renowned speakers from diverse fields of life sciences and managing the sessions smoothly. Thank you.
Will you make one favor to me?
Yes, sir.
I will like that one of my slides, if you project, which I delivered a lecture, which is question answer event in the Mendel celebrations, bicentennial birthday celebration of Mendel. Actually, you can show that slide to the audience, to the research fellows, teachers, because that is important. I could not manage it from here, but you can show that particular slide to the audience. That is my discussion. That is the question answer with a lady during the Mendel celebrations. That is very important. I was the only Indian who have participated in this function. So if possible, please run that particular slide for 2 minutes. It is only of 2 minutes duration. Thank you.
The IT cell, please I would be obliged if you could put up with the request of Professor Santosh Kumar. IT cell, please.
That slide is 7th or 8th in row. That is 8th slide. Please run that particular slide.
IT cell, please do the needful.
Slide number 8. It is audio video. Could it be possible? Hello.
Is this the slide you want, sir? Professor
Number 8.
Santosh Kumar?
Number 8.
Number 8.
It is audio video. Just you pick that point and it will start.
Sir, we are actually running late.
Okay.
So I would request you that we will try to show it to them later if possible, if you give your permission.
Okay. Thank you. Okay.
Thank you, sir. Thank you so much. Thank you so much for sharing your knowledge with all of us. Thank you.
Thank you, sir.
Thank you, sir.
Okay,
Thank you very much, sir.